Encephalitis is inflammation of the brain. There are several causes, including viral infection, autoimmune inflammation, bacterial infection, insect bites and others. Sometimes there is no known cause. Encephalitis may cause only mild flu-like signs and symptoms, such as a fever or headache, or no symptoms at all. Sometimes the flu-like symptoms are more severe. Encephalitis can also cause severe symptoms including confusion, seizures, or problems with movement or with senses such as sight or hearing. In some cases, encephalitis can be life-threatening. Prompt diagnosis and treatment are important because it's difficult to predict how encephalitis will affect each individual. Symptoms. Most people with viral encephalitis have mild flu-like symptoms, such as headache, stiff neck, fever, aches in muscles or joints, fatigue or weakness. Sometimes the signs and symptoms are more severe, and might include confusion, agitation or hallucinations, seizures, loss of sensation or being unable to move certain areas of the face or body, muscle weakness, problems with speech or hearing, loss of consciousness, including coma. 233. In infants and young children, signs and symptoms might also include bulging in the soft spots, fontanelles, of an infant's skull, nausea and vomiting, body stiffness, poor feeding or not waking for a feeding, irritability. Causes. The exact cause of encephalitis is often unknown. Autoimmune inflammation, viral and bacterial infections, and non-infectious inflammatory conditions all can cause encephalitis. There are two main types of encephalitis. Primary encephalitis. This condition occurs when a virus or other agent directly infects the brain. The infection may be concentrated in one area or widespread. A primary infection may be a reactivation of a virus that had been inactive after a previous illness. Secondary encephalitis. This condition results from a faulty immune system reaction to an infection elsewhere in the body. Instead of attacking only the cells causing the infection, the immune system mistakenly attacks healthy cells in the brain. Also known as post-infection encephalitis, secondary encephalitis often occurs 2 to 3 weeks after the initial infection. Common viral causes. The viruses that can cause encephalitis include herpes simplex virus, HSV. Both HSV type 1, associated with cold sores and fever blisters around your mouth, and HSV type 2, associated with genital herpes, can cause encephalitis. Encephalitis caused by HSV type 1 is rare but can result in significant brain damage or death. 234. Other herpes viruses. These include the Epstein-Barr virus, which commonly causes infectious mononucleosis, and the varicella-zoster virus, which commonly causes chickenpox and shingles. Enteroviruses. These viruses include the poliovirus and the Coxsackie virus, which usually cause an illness with flu-like symptoms, eye inflammation and abdominal pain. Mosquito-borne viruses. These viruses can cause infections such as West Nile, La Crosse, St. Louis, Western equine and Eastern equine encephalitis. Symptoms of an infection might appear within a few days to a couple of weeks after exposure to a mosquito-borne virus. Tick-borne viruses. The Powassan virus is carried by ticks and causes encephalitis in the Midwestern United States. Symptoms usually appear about a week after a bite from an infected tick. Rabies virus. Infection with the rabies virus, which is usually transmitted by a bite from an infected animal, causes a rapid progression to encephalitis once symptoms begin. Rabies is a rare cause of encephalitis in the United States. Childhood infections. Common childhood infections, such as measles, rubeola, mumps and German measles, rubella, used to be fairly common causes of secondary encephalitis. These causes are now rare in the United States due to the availability of vaccinations for these diseases. Risk factors. Anyone can develop encephalitis. Factors that may increase the risk include age. Some types of encephalitis are more common or more severe in certain age groups. In general, young children and older adults are at greater risk of most types of viral encephalitis. Weakened immune system. People who have HIV/AIDS, take immune-suppressing drugs or have another condition causing a weakened immune system are at increased risk of encephalitis. Geographical regions. Mosquito or tick-borne viruses are common in particular geographical regions. 235. Season of the year. Mosquito and tick-borne diseases tend to be more common in summer in many areas of the United States. Complications. The complications of encephalitis vary, depending on factors such as your age, the cause of your infection, the severity of your initial illness, the time from disease onset to treatment. People with relatively mild illness usually recover within a few weeks with no long-term complications. Complications of severe illness. Inflammation can injure the brain, possibly resulting in a coma or death. Other complications may last for months or be permanent. These complications can vary widely in severity and can include persistent fatigue, weakness or lack of muscle coordination, personality changes, memory problems, paralysis, hearing or vision defects, speech impairments. 236. Prevention. The best way to prevent viral encephalitis is to take precautions to avoid exposure to viruses that can cause the disease. Try to practice good hygiene. Wash hands frequently and thoroughly with soap and water, particularly after using the toilet and before and after meals. Don't share utensils. Don't share tableware and beverages. Teach your children good habits. Make sure they practice good hygiene and avoid sharing utensils at home and school. Get vaccinations. Keep your own and your children's vaccinations current. Before traveling, talk to your doctor about recommended vaccinations for different destinations. Protection against mosquitoes and ticks. To minimize your exposure to mosquitoes and ticks. Dress to protect yourself. Wear long-sleeve shirts and long pants if you're outside between dusk and dawn when mosquitoes are most active, and when you're in a wooded area with tall grasses and shrubs where ticks are more common. Apply mosquito repellent. Chemicals such as DEET can be applied to both the skin and clothes. To apply repellent to your face, spray it on your hands and then wipe it on your face. If you're using both sunscreen and a repellent, apply sunscreen first. Use insecticide. The Environmental Protection Agency recommends the use of products containing permethrin, which repels and kills ticks and mosquitoes. These products can be sprayed on clothing, tents and other outdoor gear. Permethrin shouldn't be applied to the skin. Avoid mosquitoes. Refrain from unnecessary activity in places where mosquitoes are most common. If possible, avoid being outdoors from dusk till dawn, when mosquitoes are most active. Repair broken windows and screens. 237. Get rid of water sources outside your home. Eliminate standing water in your yard, where mosquitoes can lay their eggs. Common problems include flowerpots or other gardening containers, flat roofs, old tires and clogged gutters. Look for outdoor signs of viral disease. If you notice sick or dying birds or animals, report your observations to your local health department. Protection for young children. Insect repellents aren't recommended for use on infants younger than 2 months of age. Instead, cover an infant carrier or stroller with mosquito netting. For older infants and children, repellents with 10% to 30% DEET are considered safe. Products containing both DEET and sunscreen aren't recommended for children because reapplication, which might be necessary for the sunscreen component, will expose the child to too much DEET. Tips for using mosquito repellent with children include: always assist children with the use of mosquito repellent. Spray on clothing and exposed skin. Apply the repellent when outdoors to lessen the risk of inhaling the repellent. Spray repellent on your hands and then apply it to your child's face. Take care around the eyes and ears. Don't use repellent on the hands of young children who may put their hands in their mouths. Wash treated skin with soap and water when you come indoors. 11.4.6 LEISHMANIASIS. Leishmaniasis is caused by a protozoa parasite from over 20 Leishmania species. Over 90 sandfly species are known to transmit Leishmania parasites. There are 3 main forms of the disease. 238. Visceral leishmaniasis, VL, also known as Kala azar, is fatal if left untreated in over 95% of cases. It is characterized by irregular bouts of fever, weight loss, enlargement of the spleen and liver, and anemia. Most cases occur in Brazil, East Africa and in India. An estimated 50,000 to 90,000 new cases of VL occur worldwide annually, with only between 25 to 45% reported to WHO. It remains one of the top parasitic diseases with outbreak and mortality potential. In 2020, more than 90% of new cases reported to WHO occurred in 10 countries: Brazil, China, Ethiopia, Eritrea, India, Kenya, Somalia, South Sudan, Sudan and Yemen. Cutaneous leishmaniasis, CL, is the most common form of leishmaniasis and causes skin lesions, mainly ulcers, on exposed parts of the body, leaving lifelong scars and serious disability or stigma. About 95% of CL cases occur in the Americas, the Mediterranean basin, the Middle East and Central Asia. In 2020, over 85% of new CL cases occurred in 10 countries: Afghanistan, Algeria, Brazil, Colombia, Iraq, Libya, Pakistan, Peru, the Syrian Arab Republic and Tunisia. It is estimated that between 600,000 to 1 million new cases occur worldwide annually. Mucocutaneous leishmaniasis leads to partial or total destruction of mucous membranes of the nose, mouth and throat. Over 90% of mucocutaneous leishmaniasis cases occur in Bolivia, the pluractional state of Brazil, Ethiopia and Peru. Transmission. Leishmania parasites are transmitted through the bites of infected female phlebotomine sandflies, which feed on blood to produce eggs. The epidemiology of leishmaniasis depends on the characteristics of the parasite and sandfly species, the local ecological characteristics of the transmission sites, current and past exposure of the human population to the parasite, and human behavior. Some 70 animal species, including humans, have been found as natural reservoir hosts of Leishmania parasites. Major risk factors. Socioeconomic conditions. Poverty increases the risk for leishmaniasis. Poor housing and domestic sanitary conditions, such as a lack of waste management or open sewerage, may increase sandfly breeding and resting. 239. Sites, as well as their access to humans. Sandflies are attracted to crowded housing as these provide a good source of blood meals. Human behavior, such as sleeping outside or on the ground, may increase risk. Malnutrition. Diets lacking protein, energy, iron, vitamin A and zinc increase the risk that an infection will progress to a full-blown disease. Population mobility. Epidemics of both cutaneous and visceral leishmaniasis are often associated with migration and the movement of non-immune people into areas with existing transmission cycles. Occupational exposure as well as widespread deforestation remains important factors. Environmental changes. The incidence of leishmaniasis can be affected by changes in urbanization and the human incursion into forested areas. Climate change. Leishmaniasis is climate sensitive as it affects the epidemiology in several ways. Changes in temperature, rainfall and humidity can have strong effects on vectors and reservoir hosts by altering their distribution and influencing their survival and population sizes. Small fluctuations in temperature can have a profound effect on the developmental cycle of Leishmania promastigotes in sandflies, allowing transmission of the parasite in areas not previously endemic for the disease. Drought, famine and flood can lead to massive displacement and migration of people to areas with transmission of Leishmania, and poor nutrition could compromise their immunity. Diagnosis and treatment. In visceral leishmaniasis, diagnosis is made by combining clinical signs with parasitological or serological tests such as rapid diagnostic tests. In cutaneous and mucocutaneous leishmaniasis 240. Serological tests have limited value and clinical manifestation with parasitological tests confirms the diagnosis. The treatment of leishmaniasis depends on several factors including type of disease, concomitant pathologies, parasite species and geographic location. Leishmaniasis is a treatable and curable disease, which requires an immune-competent system because medicines will not get rid of the parasite from the body, thus the risk of relapse if immune suppression occurs. All patients diagnosed as with visceral leishmaniasis require prompt and complete treatment. Detailed information on treatment of the various forms of the disease by geographic location is available in the WHO Technical Report Series 949, Control of Leishmaniasis. Prevention and control. Prevention and control of leishmaniasis requires a combination of intervention strategies because transmission occurs in a complex biological system involving the human or animal reservoir host, parasite and sandfly vector. Key strategies for prevention are listed below. Early diagnosis and effective prompt treatment reduces the prevalence of the disease and prevents disabilities and death. It helps to reduce transmission and to monitor the spread and burden of disease. Currently there are highly effective and safe anti-leishmanial medicines particularly for visceral leishmaniasis, although they can be difficult to use. Access to medicines has significantly improved thanks to a WHO-negotiated price scheme and a medicine donation program through WHO. Vector control helps to reduce or interrupt transmission of disease by decreasing the number of sandflies. Control methods include insecticide spray, use of insecticide-treated nets, environmental management and personal protection. Effective disease surveillance is important to promptly monitor and act during epidemics and situations with high case fatality rates under treatment. Control of animal reservoir hosts is complex and should be tailored to the local situation. Social mobilization and strengthening partnerships, mobilization and education of the community with effective behavioral change interventions must always be locally adapted. 241. Partnership and collaboration with various stakeholders and other vector-borne disease control programmers is critical. 11.5 Venoms and allergens. Insect venom allergy. During the spring and summer months, allergy sufferers are not the only ones wary of bees, wasps and other insects. These belong to the Hymenoptera order of insects, and their stings cause painful swelling and itching in non-allergic individuals. However, the proteins in the venom also activate the immune system in allergic individuals. Allergic reactions are most frequently triggered by the stings of colony-building hymenopterans. They include bees, bumblebees, wasps, hornets and ants. In Europe, the Vespula genus is responsible for a large part of the allergic reactions. Unlike the wasps of the Dolichospila genus, Vespula wasps are carnivores. As a result, they often come into contact with human populations, for example, people eating outdoors. In the US and South and Central America, fire ants are the cause of many of the reactions. European wood and garden ants only have a very small stinger, which is why their stings seldom lead to allergic reactions. Prevalence. Insect stings can trigger local allergic reactions around the puncture site or reactions with symptoms that appear entirely elsewhere in the body, such as respiratory or circulatory problems. Doctors refer to this as a generalized or systemic reaction. Researchers believe that 0.3 to 7.5% of the European population suffers from systemic allergic reactions to insect stings. Depending on the region and occupation. Since the risk increases with the frequency of the stings, beekeepers are among those most often affected. According to data from the Anaphylaxis Registry, insect stings are one of the main triggers of anaphylactic shock in adults. Experts assume a prevalence of between 0.8 and 5% in Germany for systemic reactions. There are currently no precise figures for local reactions. Research has shown that local reactions occur more frequently than generalized reactions. 242. Insect venom allergy in childhood and adulthood. There are no precise figures on the prevalence in childhood. However, we do know that children under 12 have a lower risk than adults of recurring generalized reactions. Adults 40 years of age and over with other conditions such as cardiovascular disease or asthma have an increased risk of experiencing severe allergic reactions to insect stings. Patients who have already had a severe reaction to insect stings should speak to their doctor about the possibility of immunotherapy. Triggers. Allergic reactions to insect venoms are triggered by proteins in the venom. 12 bee venom allergens, 6 wasp venom allergens, 2 bumblebee venom allergens and 2 hornet venom allergens have been identified so far. While bee stings occur mostly in the spring and summer, wasps are active in summer and autumn. Both species are frequently in contact with people, for example, on picnics or in flowering meadows. Hornet and bumblebee stings occur less frequently. Risk factors for severe allergic reactions to insect venom. Increasing age older than 40 years of age. Cardiovascular disease. Allergic asthma. Mastocytosis, a disease in which the body produces too many mast cells. A severe allergic reaction to an insect sting in the past. Symptoms. Insect stings can trigger the following reactions. 243. Normal local reactions with no allergic immune response, with swelling to less than 10 centimeters in diameter. This swelling usually subsides within a few hours, while the itching at the puncture site can continue for several days. Severe local allergic reactions with swelling to less than 10 centimeters in diameter. These reactions last longer than 24 hours, in some cases up to a week. Severe systemic allergic reactions with nausea, vomiting, diarrhea and, in rare cases, circulatory collapse. Non-allergic individuals can also experience severe reactions if they are stung by 50 to 100 insects at once, 10 to 50 in children. With this many stings, a dangerous amount of venom enters the body, which can cause damage to the kidneys and liver. Stings in the facial area, the lips, for example, are more dangerous than stings on the legs or arms. Diagnostic procedure. Diagnosis begins with a discussion during which the doctor asks the patient about the exact circumstances of the sting, any hereditary predisposition to allergies, and details about the reaction. It is important for the next phase of therapy to determine which insect triggered the allergic reaction and how high the risk is of additional, severe reactions. The doctor therefore performs a skin and/or blood test in order to confirm whether the patient has been sensitized, i.e. whether he or she has developed specific antibodies following a previous sting. The results of these tests do not provide evidence of an allergy, but they reveal important information about allergic events in the body. In patients with a high risk of anaphylaxis, skin tests are usually performed in hospital in order to minimize the risk of severe reactions to the test. A sting challenge test, the specific provocation of an insect sting, is only conducted on patients who have already successfully completed immunotherapy for at least 3 to 5 years. It is carried out under strictly controlled conditions and only under medical supervision. 244. Therapy. Specific immunotherapy, SIT, is available for patients with a severe insect venom allergy. Immunotherapy treats not only the symptoms but also the actual cause of the allergy. If the immune system is stimulated with the insect venom allergen for a period of at least 3 to 5 years, 95% of people with a wasp venom allergy do not experience any reaction or only a very weak reaction when stung again. For people with a bee venom allergy, 80% of those treated have full protection, the remaining 20% generally have milder reactions than before. This protection does not last a lifetime in all patients. People at very high risk should therefore consider lifelong immunotherapy. People at high risk of anaphylaxis must always carry an emergency kit with them containing an adrenaline auto-injector, a corticosteroid in liquid or tablet form, and an antihistamine in liquid or tablet form. 11.6 Blister and urtica-inducing insects. Many insects cause blisters, but many blisters are so tiny that you experience them as just little bumps. However, some insects and spiders produce distinct and often very large blisters. In the United States, the bites of the brown recluse spider, the yellow sac spider and fire ants cause characteristic blisters. Handling of blister beetles also causes eruption of blisters on the skin. The blister-causing bites of these insects and arachnids are not life-threatening except in cases of hypersensitive individuals, who may have a severe allergic reaction to the venom or toxin. Brown recluse spider. The brown recluse spider, native to the United States, is found in Midwestern and Southern states. You can identify the spider by the dark violin marking on its back. It is less than 1/2 inch long. The spider likes to hang out in undisturbed dark indoor and outdoor places. It is not aggressive, but will bite if disturbed. When you get bitten, you may not feel the bite, but after 2 to 8 hours, a small blister appears, surrounded by a red swollen area. The swollen area. 245. May become bluish-gray and enlarge to almost 3 inches. The bite from a brown recluse spider may take a long time to heal. The brown recluse spider, native to the United States, is found in Midwestern and Southern states. You can identify the spider by the dark violin marking on its back. Yellow sac spider. Yellow sac spiders live outside and inside homes. They account for most spider bites, but bites are often misdiagnosed by health care providers as brown recluse spider bites, according to the Michigan State University Extension 1. They are small spiders, 1/8 to 3/8 of an inch long with a light yellow or beige body. The spiders usually hide by day and come out at night. They bite when they become trapped between human skin and clothing or bed sheets. People often get bitten while gardening. The bite is usually painful. A blister will develop 1 to 10 hours later. The blister and redness is generally less severe than that produced by the brown recluse spider. Yellow sac spiders live outside and inside homes. The spiders usually hide by day and come out at night. Fire ants. Fire ants are 1/8 to 1/4 inch long and reddish brown. They originated in South America, but now have spread and are prevalent in the southern part of the United States. Fire ants build large mounds in sunny locations in fields and gardens. When coming in contact with people or animals, they will attack, biting the skin repeatedly. The sting of these ants is very painful and will itch before forming blisters. Although deaths are rare from fire ant bites, some people are allergic to the fire ant's venom. 246. Fire ants are 1/8 to 1/4 inch long and reddish brown. The sting of these ants is very painful and will itch before forming blisters. Blister beetle. Blister beetles range in length from less than 1/2 inch to 1 inch. They have a narrow body, which can be black, gray, metallic or striped. They usually hang out in groups and feed on ornamental plants, vegetables and alfalfa. The bodies of these beetles contain a toxin, cantharidin, which causes blisters on the skin. Wear gloves when handling these beetles or crops containing live or dead beetles. Horses become very ill and may die when they eat feed contaminated with dead blister beetles. Blister beetles range in length from less than 1/2 inch to 1 inch. The bodies of these beetles contain a toxin, cantharidin, which causes blisters on the skin. 11.7 Arthropods of forensic importance. Forensic entomology proceeds on the common observation that exposed remains present a temporary and progressively changing habitat and food source for a wide variety of organisms ranging from microbes like bacteria and fungi to vertebrate scavengers. Out of these, arthropod fauna comprises a major element of the biota and insects form the most constant, diverse and conspicuous group. These 6-legged creatures predominate the terrestrial and freshwater carrion fauna. Payne, 1965. He reported 522 species of animals from decomposing pig carcasses out of which 84% were insects. Goff et al. 1986. While studying carcass decomposition reported 140 arthropod taxa out of which 83% were insects. These insects are attracted in the first instance to the body fluids oozing from natural openings and to blood or serum escaping from the wounds. Cadavers are often fairly contested by great number of insect species. The overwhelming majority are flies and beetles. Dipteran families Caliphoridae, Sarcophagidae, Mussidae, Sepsidae, Sphaeroceridae, Piophilidae and Phoridae and Coleopteran families Hysteridae, Staphylinidae, Sylphidae, Claridae, Dermestidae and Tenebrionidae predominate the scene. 247. These insects form a complex food web within carrion. This web gets more complicated because of the fact that many species have different adult food preferences from that of their larvae. 1. Flies, order, Diptera, are often first on the scene. They prefer a moist corpse for their offspring, maggots, to feed on. The most significant types of fly include blowflies, family Caliphoridae. Flies in this family are often metallic in appearance and between 10 to 12 millimeters in length. In addition to the name blowfly, some members of this family are known as blue bottlefly, cluster fly, green bottles, or black blowfly. A characteristic of the blowfly is its three-segmented antennae. Hatching from an egg to the first larval stage takes from 8 hours to 1 day. Larvae have 3 stages of development called instars. Each stage is separated by a molting event. The forensic importance of this fly is that it is the first insect to come in contact with carrion because they have the ability to smell death from up to 10 miles, 16 kilometers away. Flesh flies, family Sarcophagidae. Most flesh flies breed in carrion, dung, or decaying material, but a few species lay their eggs in the open wounds of mammals, hence their common name. Characteristics of the fleshly are its 3-segmented antennae. They are medium-sized flies with black and gray longitudinal stripes on the thorax and checkering on the abdomen. Flesh flies, being viviparous, frequently give birth to live young on corpses of human and other animals, at any stage of decomposition, from newly dead through to bloated or decaying. Cheese flies, family Piophilidae. Most are scavengers in animal products and fungi. The best known member of the family is Piophyllocaceae. It is a small fly, about 4 millimeters, 1/6 inch long, found worldwide. This fly's larva infests cured meats, smoked fish, chesses, and decaying animals and is sometimes called the cheese skipper for its leaping ability. Forensic entomology uses the presence of Piophyllocaceae larvae to help estimate the date of death for human remains. They do not take up residence in a corpse until 3 to 6 months after death. The adult fly's body is black, blue-black, or bronze, with some yellow on the head, antennae, and legs. The wings are faintly iridescent and lie flat upon the fly's abdomen when at rest. Black soldier flies, Stratiumidae. They have potential for use in forensic entomology. The larvae are common scavengers in compost heaps, are found in association with carrion, Kin 248. Destructive pests in honeybee hives, and are used in manure management. The adult fly is a mimic, very close in size, color, and appearance to the organ pipe mud dauber wasp and its relatives. Larvae feed on decaying bodies. Some species can burrow to a depth of 50 centimeters over 4 days, important in buried bodies. 2. Beetles. Beetles, order Coleoptera, are generally found on the corpse when it is more decomposed. In drier conditions, the beetles can be replaced by moth flies. Rove beetles, family Staphylinidae. The rove beetles are elongate beetles with small elytra and large jaws. Like other beetles inhabiting carrion, they have fast larval development with only 3 larval stages. Creophilus species are common predators of carrion, and since they are large, is a very visible component of the fauna of corpses. Some adult Staphylinidae are early visitors to a corpse, feeding on larvae of all species of fly, including the later predatory fly larvae. They lay their eggs in the corpse, and the emerging larvae are also predators. Hyster beetles, family Hysteridae. Adult hysterids are usually shiny beetles, black or metallic green, which have an introverted head. The carrion-feeding species only become active at night when they enter the maggot-infested part of the corpse to capture and devour their maggot prey. During daylight they hide under the corpse unless it is sufficiently decayed to enable them to hide inside it. The adult lay their eggs in the corpse, inhabiting it in the later stages of decay. Carrion beetles, family Sylphidae. Adult Sylphidae have an average size of about 12 millimeters. They are also referred to as burying beetles because they dig and bury small carcasses underground. Both parents tend to their young and exhibit communal breeding. The male carrion beetle's job in care is to provide protection for the breed and carcasses from competitors. Skin, hide beetles, family Dermestidae. Hide beetles are important in the final stages of decomposition of a carcass. The adults and larvae feed on the dried skin, tendons and bone left by fly larvae. Hide beetles are the only beetle with the enzymes necessary for breaking down keratin, a protein component of hair. 249. 3. Mites. Many mites feed on corpses with macrochels mites common in the early stages of decomposition, while Tyroglyphidae and Orobatidae mites such as Rostrosits feed on dry skin in the later stages of decomposition. Nichrophorus beetles often carry on their bodies the mite Poecilocurus which feed on fly eggs. If they arrive at the corpse before any fly eggs hatch into maggot development is delayed. 4. Moths, order Lepidoptera, specifically clothes moths family Tinidae are closely related to butterflies. Most species of moth are nocturnal, but there are crepuscular and diurnal species. Moths feed on mammalian hair during their larval stages and may forage on any hair that remains on a body. They are amongst the final animals contributing to the decomposition of a corpse. 11.8 Insects SUCESSION on corpse and its RELTIONSHIP in determining time of death. The science of forensic entomology is based on the analysis of those insects which sequentially colonize a corpse as decomposition progresses and on the rate at which the various stages of their progeny develop. This entomological information can be useful during criminal investigations in order to following. Time of death. There are 2 basic approaches to the application of entomological data for estimating the time of death. During earlier stages of decomposition, the time elapsed since death or postmortem interval PMI may be determined by calculating the time required for a given species to reach the particular stage of development recovered from the corpse at the time of discovery. The insects involved in this approach are mostly dipterans, especially those belonging to the families Calliphoridae and Sarcophagidae. There are two main ways of using insects to determine elapsed time since death. 1. Using successional waves of insects. 2. Using maggot age and development. 250. The first method is based on the fact that a human body, or any kind of carrion, supports a very rapidly changing ecosystem going from the fresh state to dry bones in a matter of weeks or months depending on geographic region. During this decomposition, the remains go through rapid physical, biological and chemical changes, and different stages of the decomposition are attractive to different species of insects. Certain species of insects are often the first witnesses to a crime. They usually arrive within 24 hours of death if the season is suitable, i.e. spring, summer or fall in Canada, and can arrive within minutes in the presence of blood or other body fluids. Many species are involved at each decomposition stage and each group of insects overlaps the ones adjacent to it somewhat. Therefore, with knowledge of the regional insect fauna and times of carrion colonization, the insect assemblage associated with the remains can be analyzed to determine a window of time in which death took place. This method is used when the decedent has been dead from a few weeks up to a year. This second method, that of using maggot age and development, can give a date of death accurate to a day or less, or a range of days, and is used in the first few weeks after death. Maggots or larvae are immature stages of Diptera or two-winged flies. The insects used in this method are those that arrive first on the corpse. These flies are attracted to a corpse very soon after death. They lay their eggs on corpse, usually in a wound. Mode of death. A dead body having external injuries is more attractive to insects than one having none. So, depending upon the degree of degradation brought about by maggots, an entomologist may be able to suggest the possible mode of death. Another application is in the cases where death has occurred due to intake of drugs. A chemical analysis of the maggots found on the dead body can reveal the specific drugs, especially helpful when no human tissues are available for sending to the laboratory for other tests. Place of death. The deceased may have been killed at a place other than the body is found. With knowledge about the carrion fauna of an area and specific habits of species found on the cadaver, an entomologist can help to determine whether the person died at a place other than where the body has been found. Similarly, route of transport of a dead body may also be traced by using entomological data. There are numerous other special situations where entomologists can help too. 251. Solve crimes. If a body is found buried as soil, the entomological data can be helpful in determining the period intervening death and burial. 11.9 Summary. Arthropods are one of the most remarkable creatures on the earth. They have an unsurpassed diversity. Entomology, the study of insects, includes many fields. One of them is medical entomology. It is a field which directly concerns with the impact of arthropods and related insects on the physical as well as mental health of humans. 12.2 Introduction. Pest controls the regulation or management of a species defined as a pest, any animal, plant or fungus those impacts adversely on human activities or environment. The human response depends on the importance of the damage done and will range from tolerance, through deterrence and management, to attempts to completely eradicate the pest. Pest control measures may be performed as part of an integrated pest management strategy. In agriculture, pests are kept at bay by mechanical, cultural, chemical and biological means. Plowing and cultivation of the soil before sowing mitigate the pest burden, and crop rotation helps to reduce the buildup of a certain pest species. Concern about environment means limiting the use of pesticides in favor of other methods. This can be achieved by monitoring the crop, only applying pesticides when necessary, and by growing varieties and crops which are resistant to pests. Where possible, biological means are used, encouraging the natural enemies of the pests and introducing suitable predators or parasites. In homes and urban environments, the pests are the rodents, birds, insects and other organisms that share the habitat with humans and that feed on and or spoil possessions. Control of these pests is attempted through exclusion or quarantine, repulsion, physical removal or chemical means. Alternatively, various methods of biological control can be used including sterilization programmers. Physical and mechanical control. 254. The protection of crops and foods from insect attack by physical and mechanical means is the simplest and most effective method. Such measures consist of destruction of insects by mechanical means, burning, trapping, protective screens or barriers, use of high or low temperature, soaking in water and drying. 1. Hand picking. When only a few plants are infected, certain large conspicuous species may be removed from the plants by hand and destroyed just by pressing the abdomen or dipping them in kerosinized water. 2. Mechanical means. A rotary blow has been found to destroy over 96% of white grubs as well as cutworms, armyworms and other caterpillars. The cutting wheel is 32 inches in diameter and 14 inches wide and carries 16 steel blades. It operates by power takeoff from a tractor at 150 revolutions per minute. 3. Trapping. The insects may be turned into a tray by light and baits or the trap may be mobile and drawn across a field. 4. Light traps. The attraction of nocturnal insects to light is a common observation and the light traps have been invented and tested for killing crop pests. The attractiveness of an artificial light depends upon its candle power and color. A yellow light will attract 60% fewer insects than a white light of the same candle power and brightness. The insects attracted by light may be destroyed as they hit electric grid or they may be drowned in water and oil after they have been turned into the trap. 5. Bait traps. 255. Baits are a form of control whereby insects are attracted to a selected spot on which they can be easily removed. Bait traps have been found to be very effective in controlling the nocturnal insects like cockroaches, crickets, caterpillars and nocturnal moths. Applied control. Biological control. The term biological control means control of insects by several biological agencies such as parasites, predators and pathogens. The introduction of sterile males in a normal population and use of sex attractants have been reported to bring about effective biological control. De Bach 1964, 1974, Ray 1969, Huffaker 1974, etc. have given exhaustive account on biological control. The theoretical basis of the biological control depends on the existence of a natural enemy. The most essential characteristic is high searching ability. It should be borne in mind that a really effective enemy may be scarce in its native home because it regulates the host population at low level. There usually is one best enemy for each species in a given habitat, and one frequently is sufficient for complete biological control. Often, however, a second or third enemy species may add to host population regulation and may in fact be necessary to achieve satisfactory biological control. The best enemy species may differ for different host habitats. Hence there is generally no single best natural enemy extending throughout the range of a pest species. With these qualities, the introduced natural enemy by itself or in conjunction with other mortality factors is expected to prevent outbreak of the pest species, or at least to drop down major population fluctuations. The essential principle of biological control is to maintain a state of biological equilibrium of a living organism. This biological equilibrium is normally maintained by a set of forces called environmental resistance. Often the biological equilibrium of a living organism is disturbed either due to environmental or biotic factors, and consequently a particular organism multiplies and spreads in an unlimited 256 manner so as to assume the status of a pest. The inherent ability of an organism to survive and to reproduce within a given time and under optimal environmental conditions is known as biotic potential. Types of biological control. There are four main types of biological control, viz. I. Introduction of parasites. E. Predators. E. Pathogenic organisms such as bacteria, viruses and fungi, etc. And IV. Removal of fertile males and creating sterility in males by gamma radiation and introducing these sterile males in a normal population. In addition to these four main types, there is a fifth type of biological control where the indigenous parasite of the locality is being utilized for this purpose. 1. Parasites. Parasitic insects develop as larvae on or in a single host individual from eggs generally laid on, in or near the host, and usually consume all or most of the host body, killing the host and then pupate, either within or outside the host. The free-living adult parasite emerges from the pupa and starts the next generation anew by actively searching for host in which to ovipositor. They tend to attack only one host stage, i.e. eggs, larvae, or pupae, although there is also some overlapping in certain cases. Adult insects do not serve as hosts. Very often life cycles are commonly short, ranging from 10 days to 2 weeks or so in midsummer, but correspondingly longer in cold weather. In general, they all have great potential rates of increase. Hymenoptera. 257. This is the dominant order among all entomophagous insects. Over 2/3 of the cases of successful biological control of pest species have been achieved by Hymenopterous parasites. The most important and applied control, biological control. The term biological control means control of insects by several biological agencies such as parasites, predators and pathogens. The introduction of sterile males in a normal population and use of sex attractants have been reported to bring about effective biological control. De Bach 1964, 1974, Ray 1969, Huffaker 1974, etc. have given exhaustive account on biological control. The theoretical basis of the biological control depends on the existence of a natural enemy. The most essential characteristic is high searching ability. It should be borne in mind that a really effective enemy may be scarce in its native home because it regulates the host population at low level. There usually is one best enemy for each species in a given habitat, and one frequently is sufficient for complete biological control. Often, however, a second or third enemy species may add to host population regulation and may in fact be necessary to achieve satisfactory biological control. The best enemy species may differ for different host habitats. Hence there is generally no single best natural enemy extending throughout the range of a pest species. With these qualities, the introduced natural enemy by itself or in conjunction with other mortality factors is expected to prevent outbreak of the pest species, or at least to drop down major population fluctuations. The essential principle of biological control is to maintain a state of biological equilibrium of a living organism. This biological equilibrium is normally maintained by a set of forces called environmental resistance. Often the biological equilibrium of a living organism is disturbed either due to environmental or biotic factors, and consequently a particular organism multiplies and spreads in an unlimited 256 manner so as to assume the status of a pest. The inherent ability of an organism to survive and to reproduce within a given time and under optimal environmental conditions is known as biotic potential. Types of biological control. There are four main types of biological control, viz. I. Introduction of parasites. E. Predators. E. Pathogenic organisms such as bacteria, viruses and fungi, etc. And IV. Removal of fertile males and creating sterility in males by gamma radiation and introducing these sterile males in a normal population. In addition to these four main types, there is a fifth type of biological control where the indigenous parasite of the locality is being utilized for this purpose. 1. Parasites. Parasitic insects develop as larvae on or in a single host individual from eggs generally laid on, in or near the host, and usually consume all or most of the host body, killing the host and then pupate, either within or outside the host. The free-living adult parasite emerges from the pupa and starts the next generation anew by actively searching for host in which to ovipositor. They tend to attack only one host stage, i.e. eggs, larvae, or pupae, although there is also some overlapping in certain cases. Adult insects do not serve as hosts. Very often life cycles are commonly short, ranging from 10 days to 2 weeks or so in midsummer but correspondingly longer in cold weather. In general, they all have great potential rates of increase. Hymenoptera. 257. This is the dominant order among all entomophagous insects. Over 2/3 of the cases of successful biological control of pest species have been achieved by Hymenopterous parasites. The most important and numerous Hymenopterous parasites occur in the major groups commonly known as chalcids, braconids, ichneumonids and proctotropids. The chalcids. The chalcids attack species in nearly all orders of insects, the preferred ones being Coleoptera, Diptera, Homoptera and Lepidoptera, which also happen to include the bulk of our chief crop pests. The immature stages of chalcids predominantly develop in hosts' eggs or larvae or pupae, and some develop in adults of Homoptera. The encyrtids and aphelinids include many that specialize in attacking scale insects, mealy bugs, white flies, and related forms and have been so outstandingly successful in biological control. The braconids. This is a major large family of parasitic wasps. They have been used in biological control. Only an occasional rare species is hyperparasitic, hence the family is nearly entirely beneficial. Parasitism may be external or internal, solitary or gregarious. Their host preferences are mainly Lepidopterous and Coleopterous larvae but also commonly Diptera, including the damaging fruit flies and Homoptera, particularly aphids. The ichneumonids. The ichneumonidae comprise about 20% of all parasitic insects. Most are primary parasites, hence the family is predominantly beneficial. Some of the largest and most conspicuous parasite species occur in this family. Ichneumonidae probably parasitize wood and stem-boring larvae in the Lepidoptera, Coleoptera or Hymenoptera, but many attack Lepidopterous pupae too. Some species have a very wide host range. Most of the Triphoninae are solitary parasites of sawflies. The Ophininae are predominantly internal parasites of Lepidopterous larvae, although a few develops in the grubs of root-feeding scarabs in the soil. 258. The Serphoids. This contains several families, the most important of which are the Platygasteridae and the Scelionidae. All are parasitic on immature stages of other insects, and they are predominantly primary internal parasites. Diptera. In addition to tachnids, which comprise the most important Dipterous family in biological control, this order contains several parasitic families including Certidae, Nemestrinidae, Pipunculidae and Canopidae, etc. However, tachnids constitute the major parasitic family both in numbers of species and in economic importance, and have been the only group utilized extensively in biological control. Predators. Predator insects differ from parasitic ones in that the larvae or nymphs, as the case may be, require several to many prey individuals to attain maturity. The adults generally deposit their eggs near the prey population, and after hatching the active mobile immature search out and consume prey individuals. This need of larvae or nymphs to search is an important distinction from parasitic forms whose larvae develop on a single host individual and thus have the advantage of not having to discover additional hosts. Adults of many species are also predatory. There may be one or several generations to one of the prey. Larvae and nymphs as well as corresponding adults may be predaceous, or only one stage may exhibit the habit. Coleoptera. The main predaceous families of the Coleoptera are Coccinellidae, Staphylinidae, Hysteridae, Lampyridae, Claridae, Cantharidae, Meloidae, Cicindelidae, Carabidae, Didicidae and Gyrinidae. The Coccinellidae and the Carabidae are of most important in biological control of agricultural pests. 259. Neuroptera. Most of the species of the order Neuroptera are predatory. The most important families in biological control have been the Chrysopidae and Hemerobiidae, which attack many agricultural pests including scale insects, mealy bugs, aphids, white flies, mites, etc. Hymenoptera. About 1/4 of the families of this order is strictly predaceous. Most of the predatory forms tend to be social and live in colonies. The colonial vespid, wasps provision their paper nests with the bodies of caterpillars and other soft-bodied insects. Species of Polistes have been used in several biological control projects. Diptera. A few families are entirely predaceous and many others contain predatory species. The more common or important economic families include the Serpidae, Acylidae, Cycidomyidae, Anthemidae, Caliphoridae and Sarcophagidae. Of the latter four, many are predaceous in the egg masses of grasshoppers. Odonata. The dragonflies and damselflies are predaceous both as nymphs and as adults. The nymphs are all aquatic and feed on a wide range of aquatic insects. Biological control has been very successful in certain cases where injurious insects have become established in land they did not previously inhabit and have developed into persistent pests. A classical example of such a biological control is that of the cottony cushion scale, Eisseria purchasii in California, by a predatory coccinellid beetle, Rhodelia cardinalis, introduced from Australia. Introduction of this beetle in California resulted in almost complete and permanent destruction of Eisseria within less than 5 years. This species was so completely destroyed that the survival became numerically insignificant. Subsequently, the predator achieved complete biological. 260. Control of the cottony cushion scale in 25 additional countries and a substantial degree of control in 4 other countries. Many outstanding successes in the biological control of serious coccid pests have since been recorded. Some notable cases include the complete biological control of the Citrophilus mealybug, Pseudococcus fragilis in California, the coconut scale, Aspidiotus destructor in the island of Fiji, Maritius and Principe, Green's mealybug, Pseudococcus citriculus in Israel, the red wax scale, Ceroplasts rubens in Japan, and coffee mealybug, Planococcus kenyae in Kenya. A mention may be made of the vertebrate predators like fishes, frogs, reptiles, insectivorous birds and mammals, etc. which are the regular feeders and deserve to be provided with suitable conditions to grow and breed profusely. 3. Pathogens. Pathogenic microorganisms attack insects and have life cycles more or less characteristic or similar microorganisms developing in other groups of animals. Insects are probably subject to as wide as variety of diseases are the vertebrates. Except for the fungi, disease organisms gain entry in the host via mouth or the digestive tract, i.e. the insect host must eat plant or other food contaminated with pathogen. In case of fungi, entrance is gained through the insect integument and free water or very high humidity is generally required. Thus, fungi tend to be restricted to moist environments. However, fungi do have advantage of attacking sucking insects which because of the nature of their feeding on sap, tend to be fairly free of disease caused by microorganisms because they rarely ingest them. Virtually no insect disease organisms occur in mammals and none have been recorded from man. Thus they are safe to use in biological control even in large-scale microbial spraying operations. The most common disease of insects is caused by bacteria, fungi, viruses, and protozoa. Bacteria. 261. Because it can be produced fairly easily on a large scale and has a wide spectrum of activity, Bacillus thuringiensis was of the first microorganisms to be incorporated into a commercial, microbial insecticide. The most sensitive insects appear to be Lepidopterous caterpillars, particularly those in which alkaline conditions prevail in the midgut. This is related to the fact that the spores, it is a crystal-bearing spore former and can be conveniently stored and applied in the resistant state without loss of potency, can only enter the insect on ingestion and that the proteinaceous crystals of toxin is only liberated at around pH 9 to 10. Those pathogenic bacteria which do not form spores have proved to be of little practical value in the field and indeed only two other groups of spore formers have been employed with any success, i.e. Bacillus popilii and Bacillus cereus. The former species produces a condition known as milky disease in the Japanese beetle, Popillia japonica, and has been used extensively in North America for control of this pest and chaffer beetles. Although Bacillus cereus does not produce a crystalline toxin, it also requires closely defined midgut conditions with an optimum around pH 7. This bacillus is active against the codling moth and certain hymenopterous pests. Viruses. The classic example of the employment of viruses in pest control is the highly successful use of polyhedrosis viruses against the pine sawfly, Neodyprion certifer in Canada. A related pest Neodyprion sanii and the spruce sawfly, Dyprion hercyniae, are similarly controlled by a polyhedrosis virus. Another example is the control of alfalfa caterpillar, Calea ceretheme, by introduction of polyhedrosis virus. The cabbage looper, Trichopluginae, and other brassica caterpillars have been controlled in a similar way. Only one other group of viruses, the granulosis type, has been used against pests in field, in this case the lepidopterans, Pieris brassicae and Pieris rapi. Further, for nucleopolyhedroviruses, which are used for the control of insect pests. Fungi and protozoa. 262. Pathogenic fungi have several properties which should make them ideal organisms for use in biological control. They produce highly resistant stages and are capable of prolific spore production once they became established. However, their great dependence on microclimatic conditions, especially humidity, has seriously curtailed their use and only a few have been employed successfully. Of these, Entomophoraxeciolus has produced promising results against the spotted alfalfa aphid, Therioaphis maculata. Many protozoa appear to cause chronic rather than acute symptoms in their victims, and the Microsporidian, Philohania hyphantrae, takes up to 4 weeks to exert its full effect, which has been shown to cause mortality in the fall webworm, Hyphantraea cunea. 4. Genetic methods. Genetic pest control implies the manipulation and use of genetic material in a manner injurious to pest insects. The control of insect pest populations by the release of sterile males has been demonstrated with at least 5 insect species. This spectacular technique has been termed autocedal control and involves using an insect species to bring about its own self-destruction. It is accomplished by irradiating laboratory-reared males of the species to an extent sufficient to disrupt the genetic function of the sperm nucleus but not appreciably interfere with the normal ability of the male to mate or of the sperm to penetrate the egg of the female. However, such fertilized eggs fail to develop, so a wild female mated with a sterilized male produces no progeny. If sterile males are released in large enough number in relation to the wild population, they will fertilize more females than will the wild males. The advantage is cumulative in each generation, hence eradication may be achieved within a few generations under ideal conditions. The major success thus far has been with the screwworm, which was eradicated from the island of Curacao, from Florida, and from Texas to California by massive annual release of sterile males. A low infestation of the oriental fruit fly was eradicated from Guam in 1963. The melon fly was eradicated from Rhode Island in 1963 using a combination of a bait spray and sterile insect release, and the field cockchafer eradicated from a 74-acre plot in Switzerland in 1962. 263. The boll weevil is reported to have been eradicated from a small isolated field by the release of chemically sterilized males. Chamasterilants have been under intensive investigation but are not as yet practical. They offer the advantage of treating the wild population directly and thus avoiding the mass rearing necessary with gamma radiation. Use of pheromones. The ability of an insect to locate a mate and to copulate once it has been found is often directed by natural specific chemicals produced by the insect called pheromones. Natural chemical attractants and mating stimulants have been shown to occur in many pest insects. They are often effective in incredibly small concentrations. One caged virgin female of the introduced pine sawfly attracted over 11,000 males. Some have been chemically identified and even synthesized. Its high specificity for particular target insects, however, makes this approach highly desirable. Such phenomena gave rise to the intriguing idea of using these natural or artificial pheromones to attract and trap insects, to lure them to contact poisons, chemosterilants or pathogens, and to mask the location of females by saturating the environment with synthetic sex pheromones. Such techniques could offer highly specific control methods with little or no ecological side effects. An example of possibility is furnished by the eradication of the oriental fruit fly on Rhode Island, where the artificial pheromone or attractant viz. methyl eugenol lured the male flies to a poison-coated surface. A similar but more important achievement was the eradication of the Mediterranean fruit fly from about 1 million acres in Florida in 1956-57. It appears that adequate control from pheromones or attractants probably can only result if they are so highly effective as to be able to reduce the pest population to low level in the treated area and are used over a large area so that immigration of the pest from the outside would not negate the effort. Pheromone traps are commonly used for controlling various moths, Heliothis, Spodoptera, lucinodes, etc. 264. Chemical control. Recent discoveries of new synthetic insecticides have sparked exciting advances and major breakthroughs in the control of insect enemies. Chemicals have subdued pests that once caused widespread crop destruction, death of domestic animals, and epidemics of insect-borne human diseases. The modern insecticides are both effective and reliable. The whole world is resorting to them more and more for the solution of many insect problems. The chemicals which kill the insects by their chemical action are termed as insecticides. They are used for the protection of men, domestic animals, crops, agricultural products from the attack of insects when other methods fail to control the pests. Insecticides are seldom used in full strength but are formulated in a variety of ways to make them easier for application. Formulations of insecticides. Following are the common formulations of insecticides viz. 1. Dusts. 2. Granular formulations. 3. Insecticide fertilizer mixtures. 4. Wettable powders. 5. Solutions. 6. Emulsifiable concentrates. 7. Aerosols. 8. Fumigants, and 9. Miscellaneous formulations. Whatever may be the formulations, the poisonous chemical present in an insecticide must penetrate the vital organs and tissues of the insect and ultimately kill it. 1. Dusts. Insecticidal dusts are those powders which are used dry and mixed with or impregnated with certain organic materials or pulverized minerals, powders such as talc, pyrophyllite, bentonite 265, etc. These minerals are called carriers or vehicles since they carry the insecticide. Dusts are blown to deposit on plants by dusting machinery or blowers. Ground to a fine size, most dust will pass through a 325 mesh screen and range in size from 1 to 40 mu. The finished dust may be 0.1 to 25% active material. In dust form, in general, the toxicity of an insecticide increases as the particle size decreases. 2. Granular formulations. These are similar to dusts except for larger particle size. The range of particle size in a granular product is designated by a 2 figures mesh classification, e.g. 30/60ths means that virtually all the insecticide granules will pass through a standard 30 mesh sieve while a negligible quantity will pass through a standard 60 mesh sieve. Some of the common granular formulations are 16/30ths, 20/40ths, 24/48ths, and 30/60ths. Granular insecticides are generally used as dressings on or in the soil and may be applied with fertilizer spreaders or special granule applicators. 3. Insecticide fertilizer mixtures. Insecticide fertilizer mixtures may be formulated by adding granular insecticides to commercial fertilizers or by spraying insecticides directly onto the fertilizer. Such mixtures are applied at the regular fertilizing time to provide both plant nutrients and control of soil insects. 4. Wettable powders. These are similar to dusts but they are meant to be diluted and suspended in water and used as spray. To make an insecticidal dust act in this manner, a dispersing and wetting agent is added to the formulation. They are more concentrated than dust as they may contain as high as 75% toxicant. 5. Solutions. 266. Many of the modern synthetic insecticides are insoluble in water but soluble in organic solvents. These soluble insecticides in solution form are used directly for insect control. They are however seldom used on plants because of their phytotoxic reaction. 6. Emulsifiable concentrates. The most common and versatile formulation is the emulsifiable concentrate. This formulation consists of an insecticide, a solvent for the insecticide, and an emulsifying agent. Mixing the concentrate with water forms an emulsion. The solvent used may evaporate quickly after spraying, leaving a deposit of toxicant after the water has evaporated. The use of an emulsifying agent serves several purposes. It makes possible the diluting of a water-insoluble chemical with water. It reduces the surface tension of the spray, thus allowing it to spread and wet the treated surface more effectively, helps the spray and make a better contact with the insect cuticle. Generally, oil-soluble emulsifying substance is used. Normally, emulsions are unstable and break up into their component parts. This action is termed breaking. For spraying on plants, a quick breaking mixture is preferred, since this results in heavier deposits of toxicant. 7. Insecticidal aerosols. Aerosols are minute particles suspended in air or fog or smoke. The diameter of these particles ranges from 0.1 to 50 micro. The dispersion of insecticide into aerosol form may be accomplished by burning, vaporizing with heat, atomizing mechanically, or releasing through a small hole an insecticide that has been dissolved in a liquefied gas. The last method of aerosol preparation gives the popular aerosol bomb. 8. Fumigants. Insecticides used in the gaseous form are known as fumigants. Fumigants are most often formulated as liquids under pressure and are held in tanks. When the liquid is released in open air, it changes back to a gas. Quite often fumigants are a mixture of 2 or more gases. 267. 9. Miscellaneous formulations. There are certain insecticides which are special formulations meant for specific uses. Often insecticides are stuffed in large pills and capsules and introduced into the stomach of animal. Insecticides may be mixed in shampoos, intended for use on house pets. Waxes for use on floors may contain an insecticide. Poison baits consist of toxicants combined with foodstuff attractive to the insect pests. All these types of insecticides are special formulations designed for special purposes. Synergies. Some chemicals have the property of greatly increasing toxicity of certain insecticides. When the increased toxicity is markedly greater than the sum of the two used separately, it is termed a synergistic action. Most synergies have been used with pyrethrum or alethrin, etc. Synergistic action is important because it provides a means for a more effective insecticide and it reduces the cost of control. 12.4 Cultural control, agronomic practices. By cultural control, it is meant those methods of planting, growing and harvesting a crop which will prevent or lessen insect damage. This method consists of a slight departure from the usual time of planting, sowing or harvesting the crops or changing the plan of crop rotation, weed control, disposal of the crop remnants after harvest. Planting good seeds and resistant varieties are very important in controlling some insect pests. Since cultural methods are usually economical, they are especially useful against pests of low unit value crops. Practices which reduce the chances of buildup of pest populations may hold them below the level which will cause economic damage. Such methods are particularly applicable to field crops and forests. Knowledge of the life history or bionomics of a pest species is essential to the effective use of cultural control methods. The principle of the weakest link, or most vulnerable part of the life cycle usually applies. The environment is changed by altering 268. Farming practices at the correct time so as to kill the pests or to slow down their multiplication. In this way, the method is aimed more at prevention than at cure. If the environment is unfavorable, the pest may not reach a population level which will cause serious damage. Cultural controls are often used when chemical or biological methods have not yet been devised for an injurious species. Cleanup of the sources of infestation and changes in the planting or harvesting time are particularly important when no effective method of killing the pest is known. 1. Control by planting pest-resistant varieties. Seeds of healthy plants, preferably of a resistant variety only, should be sown as far as possible. Certain varieties are naturally resistant or less susceptible to insect attack, e.g. certain varieties of wild apples are immune to aphid attacks while most of the cultivated varieties are very susceptible. Hence, by interbreeding wild with cultivated variety, a strain can be evolved which yield good fruits and shall also will be immune to pests. Similarly, the Indian cotton Gossypium arboreum is much more resistant to the attack of jacids, whiteflies and the pink bollworms than the American cotton, G. hirsutum. Also, the indigenous cotton is resistant to leaf roller but susceptible to pink bollworm, and the foreign variety behaves in an opposite manners. Hence, by crossing the Indian species with the American species, a new strain, immune to aphids, jacids and pink bollworms, etc., can be developed. 2. Tolerance to insect infestation. A variety may be infected by insects yet survive and show less injury than others because of its ability to replace injured parts such as leaves and rootlets. 3. Plowing in relation to insect control. The plow, if used at right time, is a good tool for combating many insect pests. It disturbs or kills them, eradicates weeds upon which they might feed and breed, exposes them to natural enemies or to weather control, and buries them so deeply that few adults can emerge. A thorough stirring of the soil before planting is an indirect method for controlling the corn root aphids. 269. Because it breaks up ant colonies, kills many aphids, and prevents the growth of weeds on which they live until corn roots are available. 4. Clean cultivation. Clean cultivation means the removal of weeds, plant residues, and other materials from the fields and growing only healthy crops. The destruction of crop residues is a very good preventive measure for controlling certain species of insects, e.g. caterpillars or beetles. The elimination of remaining effectively stops further insect breeding by cutting of their food supply and shelter. Many insects infesting crops will develop on weeds which may or may not be related to these crops botanically. Hence, weed control is a good practice for reducing insect infestation. Damage due to leptocoryza is very serious when paddy fields are surrounded by weeds which provide a good shelter to the pests till the ears have been formed in them. Similarly, various species of red hairy caterpillar, Amsactomore, Amsacticolaris, and other species of this insect are prolificous and lay eggs on a number of weeds, and on emergence the caterpillars feed on the weeds and then migrate to the nearby fields of any crop. 12.4.1 Crop rotation. Crop rotation is effective as preventive measures against insects that feed on relatively few plant species or where the insects are incapable of long-distance migration, but crop rotation is useless against a general feeder. If one and the same crop is grown every year, the insect population is bound to increase due to abundance of food material. On the contrary, if crops are grown in rotation or alternate years, e.g. a crop of one plant family followed by that of different family, e.g. barley grown in spring may be followed by legumes or pulses and then wheat in winter, and so on. The crops of the same family, e.g. cotton, and lady's finger okra belonging to the same family Malvaceae must not follow same year. Mixed crops are sometimes useful as pests do not thrive in large number. 12.4.2 Tillage practice. 270. Tillage, in agriculture, the preparation of soil for planting and the cultivation of soil after planting. Tillage is the manipulation of the soil into a desired condition by mechanical means. Tools are employed to achieve some desired effect, such as pulverization, cutting, or movement. Soil is tilled to change its structure, to kill weeds, and to manage crop residues. Soil structure modification is often necessary to facilitate its intake, storage, and transmission of water and to provide a good environment for seeds and roots. Elimination of weeds is important, because they compete for water, nutrients, and light. Crop residues on the surface must be managed in order to provide conditions suitable for seeding and cultivating a crop. If the size of the soil aggregates or particles is satisfactory, preparation of the seedbed will consist only of removing weeds and the management of residues. Unfortunately, the practices associated with planting, cultivating, and harvesting usually cause destruction of soil structure. This leaves preparation of the seedbed as the best opportunity to create desirable structure, in which large and stable pores extend from the soil surface to the water table or drains, ensuring rapid infiltration and drainage of excess or free water and promoting aeration of the subsoil. When these large pores are interspersed with small ones, the soil will retain and store moisture also. Seedbed preparation procedures depend on soil texture and the desired change in size of aggregates. In soils of coarse texture, tillage will increase aggregate size, provided it is done when only the small pores are just filled with water. Tillage at other than this ideal moisture will make for smaller aggregates. By contrast, fine-textured soils form clods. These require breakage into smaller units by weathering or by machines. If too wet or too dry, the power requirements for shattering dry clods or cutting wet ones are prohibitive when using tillage alone. Thus, the farmer usually attempts tillage of such soils only after a slow rain has moistened the clods and made them friable. Some soils require deepening of the root zone to permit increased rate of water intake and improved storage. Unfavorable aeration in zones of poor drainage also limits root development and inhibits use of water in the subsoil. Tillage, particularly conventional plowing, may create a hardpan, or plow sole, that is, a compacted layer just below the zone disturbed by tillage. Such layers are more prevalent with increasing levels of mechanization. They reduce crop yields and must be shattered, allowing water to be stored in and below the shattered zone for later crops. Tillage is also associated with the loss of fertile topsoil through erosion, a serious threat to the longevity of arable land. 271. Primary tillage equipment. Equipment used to break and loosen soil for a depth of 15 to 90 centimeters, 6 to 36 inches may be called primary tillage equipment. It includes moldboard, disc, and rotary, chisel, and subsoil plows. The moldboard plow is adapted to the breaking of many soil types. It is well suited for turning under and covering crop residues. There are hundreds of different designs, each intended to function best in performing certain tasks in specified soils. The part that breaks the soil is called the bottom or base. It is composed of the share, the landside, and the moldboard. When a bottom turns the soil, it cuts a trench, or furrow, throwing to one side a ribbon of soil that is called the furrow slice. When plowing is started in the middle of a strip of land, a furrow is plowed across the field, and on the return trip a furrow slice is lapped over the first slice. This leaves a slightly higher ridge than the second, third, and other slices. The ridge is called a back furrow. When two strips of land are finished, the last furrows cut leave a trench about twice the width of one bottom, called a dead furrow. When land is broken by continuous lapping of furrows, it is called flat broken. If land is broken in alternate back furrows and dead furrows, it is said to be bedded or listed. Different soils require different shaped moldboards in order to give the same degree of pulverization of the soil. Thus, moldboards are divided into several different classes, including stubble, general purpose for clay and stiff sod soil, slit, black land, and chilled general purpose. The black land bottom is used, for example, in areas in which the soil does not scour easily, that is, where the soil does not leave the surface of the emerging plow clean and polished. The share is the cutting edge of the moldboard plow. Its configuration is related to soil type, particularly in the down suction, or concavity, of its lower surface. Generally, 3 degrees of down suction are recognized: regular for light soil, deep for ordinary dry soil, and double deep for clay and gravelly soils. In addition, the share has horizontal suction, which is the amount its point is bent out of line with the landside. Down suction causes the plow to penetrate to proper depth when pulled forward, while horizontal suction causes the plow to create the desired width of furrow. 272. Moldboard plow bottom sizes refer to width between the share wing and the landside. Tractor plow sizes generally range from 10 to 18 inches, 25 to 45 centimeters, although larger, special purpose types exist. Secondary tillage. Secondary tillage, to improve the seedbed by increased soil pulverization, to conserve moisture through destruction of weeds, and to cut up crop residues, is accomplished by use of various types of cultivators, harrows, rollers, or pulverizers, and tools for mulching and fallowing. Used for stirring the soil at comparatively shallow depths, secondary tillage equipment is generally employed after the deeper primary tillage operations. Some primary tillage tools, however, are usable for secondary tillage. There are 5 principal types of harrows: the disc, the spike tooth, the spring tooth, the rotary cross harrow, and the soil surgeon. Rollers, or pulverizers, with V-shaped wheels make a firm and continuous seedbed while crushing clods. These tools often are combined with each other. When moisture is scarce and controls of wind and water erosion necessary, tillage is sometimes carried out in such a way that crop residues are left on the surface. This system is called trash farming, stubble mulch, or subsurface tillage. Principal equipment for subsurface tillage consists of sweeps and rod weeders. Sweeps are V-shaped knives drawn below the surface with cutting planes horizontal. A mounted set of sweeps provided with power lift and depth regulation is often called a field cultivator. The typical rod weeder consists of a frame with several plow-like beams, each having a bearing at its point. Rods are extended through the bearings, which revolve slowly under power from a drive wheel. The revolving rod runs a few inches below the surface and pulls up vegetative growth. Clearance of the growth from the rod is assisted by its rotation. Rod weeders are sometimes attached to chisel plows. Some control of weeds is obtained by tillage that leaves the middles between crop rows loose and cloddy. On mechanized farms, this is often accomplished with a cultivator pulled by a tractor. When a good seedbed is prepared only in the row, the seeded crop can become established ahead of the weeds. Plowing with the moldboard plow buries the weed seeds, retards their sprouting, and tends to reduce the operations needed to control them. If weed infestations become bad, they can be reduced somewhat by undercutting. 273. Minimum tillage. The use of cropping systems with minimal tillage is usually desirable, because intensive tillage tends to break down soil structure. Techniques such as mulching also help prevent raindrops from injuring the surface structure. Excessive tillage leaves the soil susceptible to crusting, impedes water intake, increases runoff, and thus reduces water storage for crop use. Intensive vegetable production in warm climates where 3 crops per year may be grown on the same land may reduce the soil to a single grain structure that facilitates cementation and poor aeration. The loosening and granulating actions of plowing may improve soil structure if the plowing is done when the moisture content is optimal.